Hello. Very good evening to the student who have joined. Can you let me know if the voice is audible to you? If my voice is audible to you, and also if you can see the presentation in the chat box. Kindly let me know if the audio is fine and also the presentation. Okay, thank you so much. We will wait till it's 6 p.m. and then we will start today's session. Please wait till 6 p.m. and then we will start the session. Okay, so I think we can start the session. Let other students join, and I will just make the call open for all. Okay. That's fine. Okay, so very good evening to the students who have joined. I hope my voice is audible and the video is visible to you. If not, please let me know in the chat box. Okay. So today it is our week 7 live session. So let me start with the introductions. So very good evening. My name is Mousumi and I'm the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Organic Chemistry in Biology and Drug Development. And today it is our week 7 live session. So in this week 7 live session, we will discuss the topics which are included in the week 8 for your course of Organic Chemistry in Biology and Drug Development. As you all have checked in the SWAM portal, and also you all know that today, that's right now, the week 8 course content is live on the SWAM portal, and you have to submit the assignment for the same soon. So today we will discuss about the week 8 course content, and this week course 8, based on this week 8 course content, I have around 20 questions for you. All the 20 questions are MCQs, which will have 4 options. You have to pick up the correct option and let me know in the chat box that A, B, C or D, which of the following options will be correct for that particular question. These type of questions will help you to understand the your weekly assignment and also the course content better, and it will also help you to solve your weekly assignments. Okay, so if there is any problem for my audio and the presentation, please let me know. So before starting the session, I would like to sum up few points. So there will be this type of problem solving sessions every week. These sessions will be on every Monday from 6 p.m. to 8 p.m. The link is same. The Google link which you have used to join the session today, it will be same for throughout the course, and it will be live on every Monday from 6 p.m. to 8 p.m. When I will conduct the session for 12 weeks, and we will have additional 1 hour of summary session where I will discuss a few more topics of the course for this course. These sessions are live on Google Meet platform. So the important thing is, if there is a network disruption from my side, do not leave the class. Okay, I will try to rejoin as soon as possible, but the students, you do not leave the class. But let me know if there is any issue with audio and the video, everything. Okay, and during the session, if you have any kind of network issue from your side, you can rejoin at the in the session. The session is open, and you can rejoin at any time, so you don't have to worry. And since I am recording the session, I have muted your audios and videos so that there is no interruption in recording the session. Okay, but the chat box is always open. If you have any doubt, any query, any question, anything, you can let me know in the chat box. I'm monitoring the chat box every now and then. So if you have any problem, any doubt, anything, just let me know through the chat box. Okay, I would answer your query as soon as possible. And if still there is any the doubt persists after the class, you can drop an email to me. I will share my email ID at the end of the session. This presentation and the class recording, it will be shared with the students after the session. Okay, so even if you have missed something because of the network issue or anything, you can check it later also. Okay, so today, as I have mentioned, it is our week 7 live session, and we are going to discuss the topics which are in your week 8 course content, because the week 8 course content is live right now, right? So let's see what are the topics of the week 8. So the week 8 course content are mainly the chemistry of cofactors and coenzymes. Okay, so the chemistry of cofactors or coenzymes are the thing which we will discuss today. So like every week, I will show you 20 MCQ question. If each question will have 4 options, and you have to identify among that 4 option which of the option is the correct one, and you have to type it in the chat box. Okay, here the chat box is open. You can see you have to type the correct option in the chat box. A, B, C or D, which of the following will be correct for a particular question. You have to write it in the chat box. Okay, after showing a question, I will wait for 2 to 3 minutes. Then I will check the chat box. I will check the answers given by you, and then I will show you what is the correct answer, and we will discuss that topic. Okay, even if your answer is wrong, you don't have to worry, because I am not going to give you any marks for answering in the session, but answering is better for your betterment, because it will help you to understand the course better. Okay, also I would understand if you answer the question, I will understand that you are following up the class or not. Okay, so please try to be attentive and try to answer the questions. Okay, so now let me start with the session, and let me show you the first question of today's session. So the first question of today's session is, it says, the problem number 1 says that identify the incorrect statement among the followings. Your options are: number A, acetyl-CoA is the thioester of coenzyme A and acetic acid. Option number B, pyruvate is the starting point of the synthesis of acetyl-CoA. Option C, pyruvate is formed by the metabolism of glycogen. And number D, TPP, or it is the thiamine pyrophosphate, participates in the acetyl-CoA formation. So there are four statements, right? So you have to identify the statement among A, B, C, or D. Which of the following is the incorrect one? Means which of the following statement is false? Again, I will repeat the options. Number A, acetyl-CoA is the thioester of coenzyme acetic acid. Number B, pyruvate is the starting point of the synthesis of acetyl-CoA. Number C, pyruvate is formed by the metabolism of glycogen. And number D, TPP or thiamine pyrophosphate participates in acetyl-CoA formation. So these four, among these four statements, you have to identify which statement is the incorrect one. I want all of you to participate and try to answer the question. Okay. I will wait for 2 minutes, then I will check the chat box and I will show you the correct answer. Let me check the chat box. Okay, in the examination, some questions, the topics will be the same. The topics I am trying to cover, the topics will be same, but the questions will be objective kind of. So you have to read up in detail theory. That is why I am showing you all the details of a particular question when I am solving. Okay. So yeah, some questions, there may be the questions from the problem solving session. The topics will be more or less same because I'm trying to cover almost all the topics in the problem solving session. So you can expect few questions from the problem solving session in the final examination. So I think I have got a few answers. Okay, so I think number C is the most given answer. Okay, let me check if number C is the correct answer or not. So the correct answer for first question will be very good. It is number C. Pyruvate is formed by the metabolism of glycogen. This statement is false. So first, let us see what is the structure of acetyl-CoA. So acetyl-CoA is basically is a thioester. Thioester means a normal ester. We know the structure of the ester. What is the structure of the ester? The structure of the ester is there will be a alkyl group or aryl group. We will have a carbon. We will have oxygen, and we will have another group. So this is called the ester, the COO group. Now thioester, what is the thioester? So in the normal ester is a combination of alcohol and acid, right? Normal acid and alcohol. And the thioester, what is the thioester? The thioester is the combination of acid and thiol. Thiol is like it's similar like alcohol, but it has a RSH. The formula of thiol is RSH, whereas the formula of your alcohol is your ROH, right? So the thioester of the coenzyme A and acetic acid is known as the acetyl-CoA. Here is the structure, as you can see here, up to this is the structure of coenzyme A, and this blue one is the your acid part, which comes from acetic acid. The acetyl coenzyme acetyl CoA is formed from the pyruvate. Okay, so let us first check what are the acetyl CoA is the very important molecule. What is the importance of acetyl CoA? So acetyl CoA is a central intermediate of the lipid metabolism. Okay, so the glucose forms the pyruvate, forms the acetyl CoA. Okay, and amino acids also help to synthesize this acetyl CoA. This acetyl CoA goes to give us the carbon dioxide, water, and energy from ATP. It forms the eicosanoids, phospholipids, triglycerides, bile salt, cholesterol, steroid hormones, and fatty acid ketone bodies mechanism. All discussed in the central form is the acetyl CoA. Now let us check. Okay, let me just erase this part so that you can understand better. Yeah. So, what are the end products of first for understanding going to the formation of acetyl CoA? Let us understand a little bit about the glycolysis endpoint. So, the glycolysis, these are the steps for the glycolysis. So, glyceraldehyde 3-phosphate forms the 1,3 bis phosphoglycerate, and there is another side reaction. The NAD and phosphate goes to NADH and proton. Then 1,3 bis phosphoglycerate goes to 3-phosphoglycerate, and ADP converts to ATP by phosphoglycerate kinase. Phosphoglycerate pyruvate goes to the pyruvate from the ATP to ATP from pyruvate kinase, and magnesium. So what is the fate of the pyruvate? Pyruvate goes to acetyl CoA, which in the there are side reactions like coenzyme A to carbon dioxide and NAD plus to NADH. And there are other mechanisms which we will discuss later. Okay, pyruvate is also converted to L-lactate from the use the enzyme lactate dehydrogenase, and the side reaction is like NAD plus is converted to NADH. Okay. So the statement number C, which says pyruvate is formed by the metabolism of glycogen, is false. It forms from the metabolism of glucose. Okay. And also the phosphophenol pyruvate is converted to pyruvate by the mechanism of pyruvate kinase enzyme, and one ADP is converted to ATP in this particular process. So very good. All of you have answered this first question correctly. Now let's move on to the second question. So problem number 2 says the roles of TPP and lipolic acid in the formation of acetyl CoA are number A, decarboxylation and transesterification, number B, transesterification and oxidation, number C, dehydrogenation and alkylation, and number D, decarboxylation and transamination. So the roles of TPP and lipolic acid in the formation of acetyl CoA you have to identify. So these are the respective roles. Okay, the first role will be for the TPP and the second role will be for the lipoic acid. Now, let me know in the chat box A, B, C, or D which of the following is the correct formation, correct roles of the TPP and lipolic acid. Number A, decarboxylation, transesterification. Number B, transesterification and oxidation. Number C, dehydrogenation and alkylation. And number D, decarboxylation and transamination. The roles of TPP and lipoic acid in formation of acetyl-CoA. Let me check the chat box now. Okay, I have few answers. It says number B, and one number D. So I have two number B and one number D. Okay. Let me check which of the following will be the correct answer, number B or number D. So the correct answer will be, it's number A. Decarboxylation and transesterification are the roles of the TPP and lipoic acid formation, in the formation of acetyl-CoA. So this is the chart you have to remember. Okay, from this table or from this reaction cycle you can answer many questions related to this acetyl CoA formation. So the first step is the formation of hydroxyl TPP and from the thiamine pyrophosphate, and here one molecule of pyruvate is converted to the hydroxythyl TPP, okay, or thiamine pyrophosphate. So first step is the TPP is converted to hydroxythyl TPP or HETPP, and here one pyruvate is converted from one pyruvate. This acetyl group is attached to this thiamine pyrophosphate, and the one carbon dioxide is getting released. Okay, so the first step is the decarboxylation step, where the TPP participates. Okay, now it is the pyruvate, the enzyme which catalyzes this first step is the pyruvate dehydrogenase. Okay. So this is the mechanism of the pyruvate dehydrogenase reaction. Decarboxylation of pyruvate occurs with the formation of hydroxyethyl TPP, which is the step 1. Now the step 2 is the transfer of 2 carbon unit of to lipoic acid in the step 2, and is followed by the formation of acetyl-CoA in step 3. So now what will happen in the step 2? This step 2, this group, this 2 carbon chain, this CH3, CH and OH, this will be transferred to the lipoic acid. Okay, the lipoic acid here it is the structure of the lipoic acid. As you can see here, this particular group will be transferred, and this transfer is catalyzed by the dehydrolipoline transacylase, and this step is the transesterification step. Okay, the transesterification step means this portion is there here. This is the thiol group, and the thiol group is forming the thioester, and the thioester, this portion is going from this group. Okay, so this is the kind of transfer of this group to this forming the thioester formation, and here is known as the transesterification, when one ester is converted to another one. Now the third step and the most important step is the conversion of coenzyme A to the acetyl CoA, and here this as you can see here, CoSH is converted to this acetyl CoA, and this means your I mean acetyl group is attached with the coenzyme A, and this is formation of another thioester, which is a transesterification process. So the third step is acetyl group is transferred to CoA, and now the fourth step is the dihydrolipomide is reoxidized. So this dihydrolipomide is reoxidized, and in the fourth step, and here one NAD plus is converted to NADH and H plus as the side reaction. So here the enzyme which is important is the dihydrolipolyl dehydrogenase. So three enzymes are very important in this whole conversion. The enzymes are pyruvate dehydrogenase in the first step. In the second step, the enzyme which is important is the dihydrolipolyl transacylase, and the third step, the dihydrolipolyl dehydrogenase. Okay, so the three steps. So what is the first step? Pyruvate loses carbon dioxide and HETPP formed, means that is the decarboxylation step. Second step is the hydroxyethyl group is transferred to lipoic acid, and the oxidation is formed to acetyl dihydrolipomide, and now acetyl group is transferred to CoA by lipoic acid, which is the transesterification step, and dihydrolipomide is reoxidized by NAD plus to NADH. Okay, and this is the step 4 of the reaction. So the roles of TPP and lipoic acid. First step, the TPP catalyzes the decarboxylation, and lipoic acid is the transesterification. Hence, the correct answer is number A. Okay. Now I will go to the next problem. So problem number 3 says, which of the following statements is true regarding folic acid biosynthesis? Your options are number A, it consists of a pyridine ring, PABA, which is para-aminobenzoic acid and aspartic acid. Number B, the conversion of dihydroteroic acid to dihydrofolic acid is catalyzed by dihydrofolate synthesis enzyme. Number C, the conversion of the dihydroteroic acid to dihydrofolic acid is catalyzed by the dihydrofolate reductase enzyme. And number D, humans can biosynthesize folic acid. So among these four statements A, B, C and D, you have to identify the true, which is one of the following is the true or the correct statement. Again, I will repeat. Number A, it consists of pyridine ring, PABA and aspartic acid. Number B, the conversion of dihydroteroic acid to dihydrofolic acid is catalyzed by dihydrofolate synthesis enzyme. Number C, the conversion of dihydroteroic acid to dihydrofolic acid is catalyzed by dihydrofolate reductase enzyme. And number D, humans can biosynthesize folic acid. Which of the following statement is true regarding folic acid biosynthesis? Again, let me know in the chat box, ABC, which of the ABC or D, which of the following is the correct answer? Let me check the chat box. Okay, I think I have few answers. Number C, number C, number A. So I have two different options as answer. One is number C and another is number A. Let me check which of the following is the correct one. So the statement which is true regarding the folic acid biosynthesis is, it is number B. The conversion of dehydroteroic acid to dehydrofolic acid is catalyzed by dehydrofolate synthesis. This statement is true regarding the folic acid biosynthesis. First, let us see what is the structure of folic acid. So, folic acid is also known as pteroyl glutamic acid. Okay. So, this structure, as you can see, there are three portions of this structure. First is the 2-amino 4-hydroxypteridine. This ring, this bicyclic heterocyclic ring, which has this two 6-member ring joined together by a bridge. Okay. And also it has a 4 nitrogen all together, 2 nitrogen per group. So, this is known as a pteridine ring. Here we have 2-amino 4-hydroxy. So here the numbering starts from here. This is 2-amino 4-hydroxy pteridine ring. It is joined with the para-amino benzoic acid. This is the para-amino benzoic acid or PABA. And then we have a glutamic acid. So the statement number 8 says it consists of a pteridine ring, PABA and aspartic acid. This statement is false. Now let us check what is the conversion. So para-amino benzoic acid and pteridine forms the dehydroteroic acid. And this conversion is catalyzed by dehydroteroid synthesis. Dehydroteroic acid is converted to dehydrofolic acid by the action of the enzyme dehydrofolate synthesis, which states is the statement number B. So the statement number B says the conversion of dehydroteroic acid to dehydrofolic acid is catalyzed by dehydrofolate synthesis enzyme. This statement is correct. Now the third statement has said that let us say, the conversion of dehydroteroic acid to dehydrofolic acid catalyzed by dehydrofolate reductase. This statement is false because the enzyme is the dehydrofolate synthesis. And now the last step is the dehydrofolic acid to tetrahydrofolic acid, which is the active form of the folic acid. This conversion is catalyzed by dehydrofolate reductase. The fourth statement, which says humans can biosynthesize folic acid, is false. We cannot biosynthesize folic acid, and this dehydrofolate reductase enzyme is basically inhibited by this trimethoprim, which is a very important antibacterial agent in bacterial folic acid synthesis, is inhibited by this antibacterial agent trimethoprim. Okay. So the correct statement was number B. Now let us go to the next statement. Problem number 4 says sulfonamide is an dash agent and act as an inhibitor of dash. Your options are number A, antimalarial. It is an antimalarial agent and is an inhibitor of protozoal DHFR. Number B, it is an antibacterial agent and inhibitor of protozoal DHFR. Number C, it is an antimalarial agent and inhibitor of E2 dehydroteroyl synthesis. And number D, it is an antibacterial agent and it is inhibitor of E2 dehydroteroyl synthesis. So which of the following is the correct answer? Sulfonamide is and which type of agent and which is the inhibitor of which enzyme? Again, I am giving you 2 to 3 minutes and then I will check the chat box. Identify the correct answer. Let me check the chat box. Okay, I have few answers which says that it is number D. But let me check if number D is the correct answer or not. Very good. Number D is the correct answer. So sulfonamide is an antibacterial agent and it is the inhibitor of E2 dehydroterol synthesis. This is the structure of sulfonamide. So sulfonamide is similar is a type of amide like thioester ester. In the ester we have oxygen, in thioester we have thiol. In the same case in the sulfonamide, in the amide we have carbonyl group, and here we have this sulfur group. Okay, and this let us understand the chemistry of sulfonamide. So it is recognized since 1932 in clinical usage in since 1935. First compounds found to be effective antibacterial agents in safe dose ranges. Chemically, it is a molecule containing sulfonamido, the sulfonamide. So to NH2 functional group attached to an aniline structurally related to para-aminobenzoic acid or PABA. This group is also present in non-antibacterial compounds like sulfoureas, benzothiazides, furosemide, and acetazolamide, and they act as antibacterial agents by inhibiting bacterial growth and activity and commonly called as sulfa drugs. So folate metabolism, pteridine, and which is dihydroteroate diphosphate plus para-aminobenzoic acid or PABA is converted to dihydroteroic acid DHP, and the enzyme catalyzes the dihydroteroate synthesis or DHPS, and sulfonamides they inhibit this particular enzyme. This this table is very important. The many questions can be given from this table, so remember this correctly. Dihydroteroic acid is converted to dihydrofolic acid by the catalysis of enzyme dihydrofolate synthesis, and dihydrofolic acid is converted to tetrahydrofolic acid with the enzyme dihydrofolic reductase, where timethoprim inhibits this particular enzyme, and it is also an antibacterial agent. Okay. So option D was the correct one. Now I will go to the next problem. Problem number 5 says which of the following enzymes converts DUMP to DTMP? Your options are number A SHT, number B DHFR, number C DHFS, and number D TS. Which of the following enzyme converts DUMP to DTMP? So whenever we are understanding about the enzymes and everything, there will be many short forms or single letter notations for the enzyme which you have to remember. Not every time the question will be given with the complete name of the enzyme. Okay, like dihydrofolate reductase, it can be given also as DHFR. Dihydrofolate synthesis can be given as DHFS. Okay, so try to remember the short forms of the enzymes also. Now you have to identify which of the following enzyme converts DUMP to DTMP. Let me check the chat box now. Okay, I have few answers. It says number D, number C, number T. So I have two different options again. One says it is number D, one says it's number C. Let us now check which of the following will be the correct one. So the correct answer will be it's number D, TS. So TS is known as the thymidylate synthetase or TS. Okay, it catalyzes the conversion of DUMP to DTMP. So as you can see, this is the formula of DUMP. Now where there is a uracil unit, and in the DTMP we have a thymine unit. Okay, so the difference between uracil and thymine is the this CH3 or methyl group in this position, right? So the enzyme which is responsible for this conversion is thymidylate synthetase, which at the side reaction happens where the N5N10 methylene tetrahydrofolate is converted to 7,8 dihydrofolate, and from here this methyl group is transferred to this position. Okay. So thymidate synthesis, this DUMP catalyzes the conversion of DUMP to DTMP, which is converted to DTDP, so DTDT, and DNA, which eventually leads to the DNA synthesis. Again, this is known as the salvage pathway, where thymidine is converted to thymidine kinase to DTMP. Here we have the de novo synthesis pathway, where the CH2 folate is converted to H4 folate, and the serine is converted to glycine by serine hydroxymethyl transferase, and also NADPH is converted to NADP by dihydrofolate reductase, and we have the H2 folate. Okay, so this pathway, if you do not remember it, it's fine, but please remember this conversion and this enzymes. Okay. So now I will go to the next formation. So yeah, SHT is the serine hydroxymethyl transferase. Okay. So problem number 6 says that reaction involved in the first step of the conversion of uracil to thymine is number A, Michael addition, number B, sigmatropic rearrangement, number C, aldol condensation, and number D, Claisen condensation. So which of the following reaction is the first step for the conversion of uracil to thymine? Michael addition, sigmatropic rearrangement, aldol condensation, or Claisen condensation? Again, and let me wait for 2 minutes, then I will check the chat box and I will show you the correct answer. Let me check the chat box now. Okay, I have few answers. One says it's number A, number D. Okay, I have two different options. One says it's number A and another is number D. Let me check which of the following is the correct answer. So the correct answer will be number A. Michael addition was the first step of the conversion of uracil to thymine. Now let us understand what is the Michael addition. So Michael reaction or Michael addition is the addition of an enolate, which is a nucleophile, to an electrophilic alkane conjugated to an electron withdrawing group. Okay. So from CH we have to form CC bond to break pi bond. So for sigma bond we have to break the pi bond. So here, for example, it's the enolate. Enolate means there we have a carbonyl and followed by an anion here, which is a nucleophile. Okay, which is usually formed via the deprotonation. And we have a electrophile which has a double bond and saturation, and it is attached with the electron withdrawing group, which is an alpha beta unsaturated compound. So now the conversion, the new CC1 formation happens like this. Nucleophile attacks here, and this negative charge is here, and this it will have the protonation, and we will have this kind of product. Okay, the electron withdrawing group can be many things like ketone, ester, nitrile, and nitro. So conjugate addition is the general name for addition of nucleophile to beta position of alpha beta unsaturated compound. Okay. Typical nucleophile for conjugation is enolate, thiols or thiolates, enamine, amine, and oligocuprates. So the conversion of uracil to thymine, which is the DUDP, so DUMP. Now first, from the DUMP is formed from DUDP, and this is converted to DTMP from the your thyminyl synthesis. So this is the complete formation. I have shown you earlier also. Here it is the complete reaction. So first we have this DHF. Okay, and DHF is converted to the THF. Okay, and the DHF is the dihydrofolic acid is converted to THF, and the enzyme is the DHFR. Okay, and here NADPH is converted to NAD plus, followed by the formation of THF to 5,10 methylene THF. Okay, and here as you can see, the particular your serine is converted to glycine, and this 5,10 methylene THF is converted to DHF again with the help of thymidine surface, and here the proton and this C is this proton methylene. Okay, so the first step is the Michael addition. Now I will go to the next slide. So problem number 7 says which co-substrate is involved in the conversion of dihydroxyproline from the proline. Your options are number A 2-oxoglutarate, number B coenzyme A, number C NAD plus, and number D GABA or okay, so GABA. So which is gamma aminobutyric acid. So which co-substrate is involved in the conversion of hydroxyproline to proline? 2-oxoglucarate, coenzyme A, NAD plus, or GABA. Let me check the chat box now. Okay, so I have few answers. Let me check. Number A is the given answer for this question. Let me check if number A is the correct one or not. So the correct answer for problem number 7 is, which says that which co-substrate is involved in conversion of hydroxyproline to proline. So the correct answer is very good. Number A, 2-oxoglutarate is the co-substrate which is involved in the conversion of hydroxyproline from proline. So we are discussing the mechanism of the hydroxylation of the proline to form 4-hydroxyproline. The ascorbic acid is acting as an antioxidant. Its role is to keep the iron in +2 state. Now the question is how this hydroxylation is carried out by this propyl hydroxylase or PHD. In addition to ferrous, this PHD enzyme requires another co-substrate which is alpha ketoglutarate or 2-oxoglutarate. What is the 2-oxoglutarate? It in presence of oxygen helps iron to form very superoxidant which is called a ferryl oxospecies where the iron is in +4 state. Now how it is formed? This is the resting state of the enzyme and the cofactor iron. So it has got 3 water molecules, an aspartate and 2 histidine ligands. Okay. So here we have proline and the 2-hydroxyproline. The co-substrate is the 2-oxoglycerate which form the succinate, and here we have the oxygen and iron in +2 state which is converted to iron in +3 state in carbon dioxide. And now this this carbon dioxide again, this reoxidation form is formed by the vitamin C or oxygen. This is acts as an antioxidant and it keeps the iron in the +2 state from the +3 state, and the dihydroascorbate is formed in this motion position. Okay, so this conversion, this chart is again also very important, and you have to remember the roles of the two oxoglycerate, the roles of the ascorbic acid in the conversion, this particular conversion. Okay, now I will go to the next point. The problem number 8 says the enzyme which catalyzes the fibrinogen to fibrin conversion is. Your options are number A trypsin, number B thrombin, number C prothrombin, and number D pepsin. So which of the following enzyme catalyzes the fibrinogen to fibrin conversion? Trypsin, thrombin, prothrombin, or pepsin? Which enzyme catalyzes the fibrinogen to fibrin conversion? So let me check the chat box now. Okay, I have few answers. So I have the answers like number B is the only given answer. Let me check if number B is the correct answer or not. The correct answer is very good. It's number B, thrombin. So prothrombin is converted to thrombin by the thrombin activators and calcium ion. This thrombin catalyzes the conversion of fibrinogen to fibrinogen monomer, which eventually forms the fibrin fiber. And calcium ion is again present here. Okay, thrombin can be also activated fibrin stabilizing factor. It forms. Okay, and this fibrin fiber forms a cross-linked fibrin fiber, which is the heart of the coagulation of the blood clots. So here, if we have any injury or there will be a platelet aggregation at that position of injury, and we will have the activation or the coagulation. So the first step is the clot initiation, and this activation of coagulation will trigger the thrombin, which will catalyze the fibrinogen to fibrin conversion. This fibrin will form the fibrin polymers, and this retraction and clot formation will happen. This fibrin, after the healing of the particular injury, the fibrin fragment, it will just dissolve, and it is the fibrinolysis. So yeah, thrombin is the correct answer, which catalyzes the fibrinogen to fibrin conversion. So now I will go to the next problem. Problem number 9 says which of the following is or are vitamin K antagonists? Your options are number A, documarol, number B, warfarin, number C, menadione, number D, both A and B. So which of the following are the vitamin K antagonists? Documarol, warfarin, menadione, or both A and B? Let me check the chat box now. Okay, I have few answers which says number D and number B. Okay, so I have number D and number B, two different options for this question. Let me check if number B is correct one or number D is correct one. So the correct answer is, it is number D. Both A and B, both decumerol and warfarin are the vitamin K antagonists. Okay, these are the structures of decumerol and warfarin. So vitamin K antagonist. So let us first understand the vitamin K cycle and its inhibition by vitamin K antagonist. So vitamin K is converted to vitamin K hydroquinone, which is converted to vitamin K epoxide and then vitamin K. So what are the enzymes which are involved? So first vitamin K to vitamin K hydroquinone is the DT diaphorase. Okay, then from vitamin K hydroquinone to vitamin K epoxide is converted by gamma glutamyl carboxylase. It converts the gluprotein to GLA protein, and vitamin K epoxide reductase. This enzyme catalyzes the formation of vitamin K epoxide to vitamin K. Now, vitamin K antagonists, they will inhibit these enzymes, this DT diaphorase and vitamin K epoxide reductase. So warfarin is the anticoagulant and it prevents the treat the thermophilic event. So why vitamin K needs to be inhibited sometimes by the vitamin K antagonist? So these basically these agents act as an anticoagulant. So whenever there is a sometimes what happens, like normal blood flow in the our veins and arteries and veins, there are coagulation of blood sometimes. So this for the main so what are the there are many some in some regions and in some diseases this happen. So the main for that the warfarin or anticoagulants are used. So main side effect are the bleeding gums, blood in stools, hematosis, uncontrollable internal bleeding. Okay, and the mechanism of action is the function of vitamin K is no synthesis and activation of clotting factors 2, 7, 9, and 10. Nursing consideration, baseline assessment, monitoring signal of bleeding, evaluating therapeutic effect, and clinical teaching is to provide a safe administration. What is the mechanism of action of decumerol? Like all 4-hydroxycoumarin drug, it is a competitive inhibitor of vitamin K epoxide reductase. Here, it is a competitive inhibitor of vitamin K epoxide reductase, an enzyme that recycles vitamin K, thus causing depletion of active vitamin K in the blood. This prevents the formation of active form of the prothrombin and the several other coagulation enzymes. These compounds are not antagonists of vitamin K directly as they are in pharmaceutical uses, but rather promote depletion of vitamin K in the bodily tissues, allowing the vitamin K's mechanism of action as a potent medication for the decumeral toxicity. The mechanism of action of vitamin K along with the toxicity of decumerol are measured in the prothrombin time or PT in the blood test. Okay, so yeah, both A and B were the vitamin K antagonists. Now I will go to the next problem. So problem number 10 says, which of the following statements is false regarding the ascorbic acid? Your options are: number A, it is a strong antioxidant. Number B, it catalyzes the hydroxylation reaction. Number C, it is reproduced inside the human body, not required through trial. And number D, it chelates with iron to make it more soluble. So which of the following statement is false regarding ascorbic acid? So now let me check the chat box. Okay, I have few answers. It says number C. Okay, let us check if number C is the correct answer or not. So the statement which is false regarding the ascorbic acid is number C. It is produced inside our body, not required to diet. This is the false statement because vitamin C is not produced in our body. We have to acquire it through the diet. So this is the structure of the ascorbic acid. So what are the signs of the deficiency of the vitamin C? Is the bleeding of the skin and mucus, fatigue and depression, loose teeth, increased susceptibility to infection, rough and dry skin, muscle weakness, joint and body ache, and nausea and stomach cramps. Okay. So it is a strong antioxidant. It catalyzes a hydroxylation reaction. It chelates with iron to make it more soluble, but it is not produced in human body. It is required to. We have to take it up with the diet. Now, problem number 11, it says, which of the following statements regarding mature prothrombin is true? Your options are number A, mature prothrombin has 10 of the glutamyl residues in the first 42 residues from amine terminus. Number B, the carboxylation process is the vitamin K independent. Number C, in the mature prothrombin, glutamyl residues are modified as gamma carboxyglutamyl residues. And number D, the gamma carboxyglutamyl residues are good chelators of magnesium ions. So, which of the following statement about regarding mature prothrombin is true? Number A, mature prothrombin has 10 of the glutamyl residues in first 42 residues from the amine terminus. Number B, the carboxylation process of vitamin K independent. Number C, the mature prothrombin and the glutamyl residues are modified as gamma carboxyglutamyl residues. And number D, gamma carboxyglutamyl residues are good chelators of magnesium plus 2 ions. Let me check the chat box now. Yes, I have your answer. It says number A, number C, and it could be A. Okay, let me say number A and number C are the given answers, I think. Let me check which of the following is the correct one. So the correct answer is it's number C. In mature prothrombin, the glutamyl residues are modified as gamma carboxyglutamyl residues. So this is the glutamate residues close to the NH terminus of the group of the preprothrombin, and the gamma glutamyl carboxylase is converts this glutamate residue to gamma carboxyglutamyl residue in the mature prothrombin. Okay, and here is the site which is site of the calcium binding. This gamma carboxyglutamyl residues are good chelators of the calcium ion, not the magnesium ion. Okay, and this step is the not vitamin K independent; it's the vitamin K independent dependent steps. Okay. So now next problem. Problem number 12 says which of the following statements is false regarding the methotrexate? Your options are number A, it's a potent anti-cancer drug. Number B, it works by catalyzing the dihydrofolate reductase. Number C, the interaction is non-covalent. And number D, the crystal structure is known. So which of the following statement is false regarding the methotrexate? So which of the following statements is false regarding methotrexate? Your options are number A, it is a potent anti-cancer drug. Number B, it works by catalyzing the dihydrofolate reductase. Number C, the interaction is non-covalent. And number D, its crystal structure is known. Let me check the chat box. Okay, I am actually a PhD scholar. Okay, I'm not a student and also not a faculty, but I am a research scholar. I am a senior research scholar in the IIT Dhanbad. Okay, I'm pursuing my research in the organic and medicinal chemistry. Okay, so I think I have few option answers for the question. So I think number C and number A, I think these are the options for this question. Let me check which of the following is the correct one for the problem number 12. So the statement which is false regarding methotrexate is it's number B. It works by catalyzing the dihydrofolate reductase. This statement is false. Okay, so this is the structure of the methotrexate. So mechanism of action, methotrexate is an antifolate belonging to the anti-metabolite class of anti-neoplastic agents. This is the cell cycle specific chemotherapeutic agent and act as a S-phase and thus inhibit the DNA synthesis. So folic acid conversion to tetrahydrofolic acid, it is by the enzyme dehydrofolate reductase, it is inhibited by methotrexate. So as you can see the structures of methotrexate and the folic acid, you will see there are few very small change difference in the structure of the methotrexate and folic acid. So thus methotrexate acts as a substrate for the dehydrofolate reductase and thus inhibiting it. So when methotrexate binds with this enzyme, folic acid will not bind to the dihydrofolate reductase enzyme, and that's how it is inhibited. Okay, so THF included the two stage of the biosynthesis of purines, adenine and guanine, and one stage synthesis of pyrimidine, thymidine, and cytosine. Okay, so remember methotrexate, it works by inhibiting the dihydrofolate reductase, not by catalyzing. It acts as a substrate because it the structure similarity of the folic acid, which is the original substrate of dihydrofolate reductase, and thus the conversion of folic acid to the tetrahydrofolic acid is inhibited, and this inhibition because of this mechanism of action, it acts as an anti-cancer agent. Okay. So now I will go to the next problem. Problem number 13 says which of the following statement is incorrect? Your options are number A, iron 2 is a cofactor. Ascorbate maintains, number B, ascorbate maintains the oxidation level of iron at +2 state. Number C, molecular oxygen accepts the electrons and is reduced to water. And number D, DTT acts as an oxidizing agent to keep the tripeptide in the thiol stage. Okay. So which of the following statement is incorrect? Number A, iron 2 is a cofactor. Number B, ascorbate maintains the oxidation level of iron at +2 state. Number C, molecular oxygen accepts the electrons and reduced to water. And number D, DTT acts as an oxidizing agent to keep the tripeptide in the thiol stage. So which of the following statement is incorrect? There may be a little bit of noise problem, so please bear with me for some time as there is a procession nearby. So the voices, my voice is audible, right? Is there is any disturbance? So please let me know. Okay, the topic of my research is organic chemistry, the synthetic organic chemistry, and also the medicinal chemistry. Okay, so I think I have few answers for the question already. So number D is said to be the incorrect one. Okay, let me check if number D is the correct one or not. Okay, just wait for some time. There is some noise coming up here. So, which of the following statement is false regarding methotrexate? Oh, this is not the next one. Okay, let me check if the number D is the correct answer or not. Yes, very good. Number D is the correct answer. DTT acts as an oxidizing agent to keep the tripeptide in thiol stage. This statement is incorrect. So, dithiotritol or DTT is commercially available chemical used to reduce the disulfide bonds made between the cysteine side chains in peptides and proteins. The overall reaction which reduces the cysteine disulfide, also called as cysteine, to free cysteine residues through sequential disulfide exchange reaction is shown below. Okay, this is a redox reaction, and the two reduction half reaction for oxidized DTT and cysteine are also shown here. Okay, the DTT plus 2, this half reaction is a DDT 1 SS bond plus the 2 electron plus 2H to DDT 2 with thiol groups. The reduction potential is minus 0.330 volt, and cysteine plus 2 electron to 2 cysteine, the potential is minus 0.3040 volt. Suppose you have a peptide contains disulfide between two cysteine residues called cysteine. You want to reduce the disulfide bond at 25 degrees centigrade with cysteine. DDT is reduced at 0.1 molar, and cysteine is with really favorable reactions for reducing the cysteine bond in your peptide. Okay, so this is the both half reaction. You don't need to remember this thing. So remember here that DDT is a commercially available agent which is used to reduce, it is a reducing agent, not the oxidizing. It is used to reduce the disulfide bonds to make the cysteine side chains in the peptides and protein. Okay. So now I will go to the next problem. So problem number 14 says glycerol to 3-hydroxypropanoic acid conversion is catalyzed by which enzymes? Your options are number A, glycerol hydratase and alcohol dehydrogenase, number B, glycerol dehydratase and alcohol dehydrogenase, number C, glycerol dehydratase and aldehyde dehydrogenase, and number D, alcohol dehydrogenase and aldehyde dehydrogenase. So which of the following is the correct answer? The glycerol to 3-hydroxypropanoic acid conversion is catalyzed by the enzymes number A, glycerol hydratase and alcohol dehydrogenase, number B, glycerol dehydratase and alcohol dehydrogenase, number C, glycerol dehydratase and aldehyde dehydrogenase, and number D, alcohol dehydrogenase and aldehyde dehydrogenase. Which of the following is the correct answer? So glycerol to 3-hydroxypropanoic acid conversion is catalyzed by which enzyme? Number A, glycerol hydratase and alcohol dehydrogenase. Number B, glycerol dehydratase and alcohol dehydrogenase. Number C, glycerol dehydratase and aldehyde dehydrogenase. And number D, alcohol dehydrogenase and aldehyde dehydrogenase. Let me check the chat box. Okay, so I have few answers for this question again. It's a number C, number B, number C, and number C. Okay, number C is the most given answer, but we also have a number B. So let me check which of the following is the correct answer. So the correct answer will be, yes, it is number C, glycerol dehydratase and aldehyde dehydrogenase. These are the enzymes which catalyzes the glycerol to T3-hydroxypropanoic acid conversion. So this is the glycerol. To first step is the glycerol to 3-hydroxypropanol, okay, which is the aldehyde. So basically, its first step is the alcohol to aldehyde formation, and here the glycerol dehydratase is the enzyme, and we also have coenzyme B12, and 3-hydroxypropanol converted to 3-hydroxypropanoic acid with the aldehyde dehydrogenase enzyme, and NAD1 NAD plus is converted to NADH in this step. Again, we have a few other steps also, like 3-hydroxypropanol is converted to 3-hydroxypropanol CoA by the propanoldehyde dehydrogenase enzyme, and here CoA is there is a CoA addition, and NAD plus is converted to NADH, and the this 3-hydroxypropanol CoA is converted to 3-hydroxypropanol phosphate by the phosphotransylase, and okay, and this 3-hydroxypropanol phosphate is converted to 3-hydroxypropanoic acid with the propanoid kinase. So this is the also another step which is a CoA dependent step. So there are two steps from glycerol to 3-hydroxypropanoic acid conversion. We have two steps. One step is the CoA independent step where the enzymes are involved is the aldehyde dehydrogenase, and another step is the CoA dependent step where there are several enzymes like propanaldehyde dehydrogenase, phosphotriacylase, propanoate kinase, etc. Okay. Now I will go to the next problem. Problem number 15 says proline to hydroxyproline conversion catalyzed by proline hydroxylase is associated with the generation of which gas? Your options are number A oxygen, number B NO2, number B carbon dioxide, number D sulfur dioxide. So proline to hydroxyproline conversion is catalyzed by proline hydroxylase and it is associated with generation of which gas? O2, CO2, O2, NO2, CO2 or SO2. Let me know in the chat box. After 2 minutes, I will check the chat box and then I will show you the correct answer and we will discuss a little bit. Proline to hydroxyproline conversion is catalyzed by proline hydroxylase is associated with generation of which gas? O2, NO2, CO2, SO2. Let me check the chat box now. Okay, so I have let's first me check me the answer. So answer C, number C is the most given answer. Okay, let's check if the number C is the correct one for problem number 15. Yeah, so I have qualified for JRF in 2000. I have qualified in I think 2020. Yeah, the net 20 in the CSR net 2020 in the December one. I have qualified. Okay, so I have qualified in the June one. Okay, the examination was in December, but it was for June. So yeah, and how many marks you need to get selected from the PhD scholar? It depends. Okay, it depends on the yearly cutoff. It changes from time to time. But for as a to select in selection of GRF, you have to score above 100. Okay, this is I think in my year the cutoff was 101 or something, but it varies from year to year and subject to subject. The year to year it varies. In chemistry it's around 100 or 101 to sometime 99 also if the question is too so tough, many tough. So it changes from year to year and examination to examination. Okay, so number C is the given answer for problem number 15. Let me check if it is correct or not. So the correct answer will be yes, it is number C. Carbon dioxide is the gas which generates when the proline is converted to hydroxyproline by the catalyze of the proline hydroxylase. So this is the proline. This is the structure of proline. This is the structure of the hydroxyproline. Okay. So now the conversion of the proline to hydroxyproline is catalyzed by your proline hydroxylase, and alpha ketoglutarate is converted to succinate here, and we have the oxygen to convert it to carbon dioxide, and iron 2 plus is here is present as a cofactor. Okay. So now I will go to the next problem. Problem number 16 says the side product formed at the time of the conversion of serine to glycine through the serine hydroxymethyl transferase or SHT is. Your options are number A, PHCHO, which is benzaldehyde, number B, the acetaldehyde, number C, formaldehyde, and number D, acetone. So, which is the side product formed at the time of the conversion of serine to glycerine through the serine hydroxymethyl transferase enzyme? Number A, benzaldehyde, number C, B, acetaldehyde, number C, formaldehyde, number D, acetone. Which is the correct answer? Let me check the chat box. Okay, so I have few answers. This is formaldehyde, number C, number C. Let me check if number C is the correct answer or not. The correct answer is yes, it's number C. Formaldehyde is the side product formed at the time of the conversion of serine to glycine. So serine hydroxymethyl transferase or SHT is the another pyridoxal phosphate dependent enzyme catalyzes the interconversion of amino acid serine with glycine and formaldehyde. So this mechanism, so this is the serine, it converts to glycine and formaldehyde. Okay. And this is the cleavage system. So there we have this glycine, we have this a particular glycine, and here we have the this is a little bit complicated mechanism. You don't need to memorize this complete mechanism. Okay, so just it's only for your information. So this is the glycine cleavage system. Okay, so in this as you can see in this glycine cleavage system, we have the glycine. Okay, and this glycine is formed with the serine, and this is the there is the enzyme. There's a carbon dioxide formation, also the formaldehyde formation also, and you don't need to remember. I think this is a little bit complex, so you don't need to remember this complex mechanism. So just remember the formaldehyde will be forming, and as you can see from the structure itself, so here this portion is forming the formaldehyde, and serine hydroxymethyl transferase is also. So you what you have to remember is it is a pyridoxal phosphate dependent enzyme. Okay, and the interconversion of serine to glycine is done by this enzyme. Okay, so now I will go to the next problem. Problem number 70 says pyruvate carboxylase uses dash as a prosthetic group. Your options are number A acetyl CoA, number B retinol, number C ascorbic acid, and number D biotin. So pyruvate carboxylase uses which as a prosthetic group? Number A acetyl CoA, number B retinol, number C ascorbic acid, and number D biotin. Pyruvate carboxylase uses which as a prosthetic group? Acetyl CoA, retinol, ascorbic acid, or biotin? Let me know in the chat box. A, B, C, or D, which of the following is the correct one? Let me check the chat box now. Okay, so I think you have many questions regarding the PhD. So I have not decided yet. Maybe I will opt for more research as a postdoc. And after that, let's see how it goes around. Okay, so number D is the given answer for the problem number 17, I think. So let me check if it is the correct one or not. The correct answer is yes, it's number D. Biotin is the use the prosthetic group for the pyruvate carboxylase. So this is the structure of the biotin. So pyruvate Carboxylase uses biotin as prosthetic group. So this is the structure of biotin. Here there is a nitrogen which is subject to carboxylation. So biotin has a 5-carbon side chain whose terminal carboxyl is an amide linkage with the epsilon amino group of the enzyme lysine. This is the epsilon amino group of enzyme lysine. The biotin and lysine side chain form a long swinging up that allows the biotin ring to swing back and forth between two active sites. The pyruvate is converted to oxaloacetate and by the enzyme pyruvate carboxylase, and it uses biotin as a cofactor. Okay, and this is the mechanism of particular mechanism. Now I will go to the next problem. Problem number 18 says the letter K in the vitamin K stands for number A kinetics, number B chymotrypsin, number C coagulation, and number D catalysis. So the letter K in vitamin K stands for which? Which of the following will be the correct answer? Number A kinetics, number B chymotrypsin, number C coagulation, number D catalysis. So the letter K in vitamin K stands for which? Let me check the chat box first. Okay, I have answer like number C, number D. Number T or number C. Let me check which of the following is the correct one. So K in vitamin K stands for yes, number C coagulation. So vitamin K helps in blood clotting, is essential to stop bleeding from the wounds. People with vitamin K deficiency may experience easy bruising, nosebleeds, etc. Although the deficits of this vitamin are rare to occur. The sources of vitamin K are animal fats like egg yolk, whole milk, red meat, and dark green vegetables like spinach and asparagus. We already learned some medicinal aspects while studying the chemistry in biological system. For example, folic acid biosynthesis inhibitors. Now, next coenzyme that we are going to discuss is called vitamin K. And this K is actually involved in the blood coagulation. Blood coagulation means whenever there is a cut in the blood, it would not come outside after few seconds or a minute. Now this K actually stands for coagulation. Now what it say in the German language, it starts with the K. There is some blackish thin called clot that covers the cut, and the clot process is dependent on vitamin K. If you have a deficiency of vitamin K, then blood clotting will be very low, so lots of blood will be lost. In addition, vitamin K is also, you need a lot of calcium ions because the clot is made up of protein which is called fibrin. The fibrin protein in presence of calcium forms cross-linking protein that makes it very insoluble mass, which is known as a clot. Okay, so vitamin K, in vitamin K, K stands for coagulation is the correct answer. Problem number 19 says, which of the following vitamins chelate with iron at its most solubility? For its most, your options are number A, vitamin A, number B, vitamin K, number C, vitamin C, number D, vitamin D. Which of the following vitamins chelates with iron 2 ions for more solubility? A, K, A, C, or B? I think all of you can answer it very correctly, so I want all of you to answer this question. Okay, let me check the chat box. Number C is the given answer from problem number 19. Okay, let me check if number C is the correct answer or not. Yes, number C means vitamin C is the correct answer. Vitamin C chelates with iron for more solubility. This is the structure of vitamin C or ascorbic acid. It is a very simple structure. So this particular vitamin C or the ascorbic acid, this has two hydroxyl groups here, which sometimes it gets deprotonated and form two anions. These anions act as a chelator of the iron. So as you can see, there is an intramolecular chelation with the iron, and for the more solubility. The benefits of chelated iron with vitamin C is boost the RBC production, absorbable form of the iron for all, suppose the cognitive performance, vitamin C to enhance the iron absorption. Okay. Sometimes the doctors say to take the vitamin C supplements and iron supplements together, or sometimes they say that to eat the iron rich fruit with some citrus fruits or lemons, because this will increase the reabsorption of the iron source, because the solubility of iron too increases with this vitamin. So the last question of today's session, it says that which of the following conversions can be termed as a reduction reaction? Your options are number A, malate to oxaloacetate, number B, beta aspartate semialdehyde to homoserine, Number C, glyceraldehyde 3-phosphate to 1,3-diphosphoglyceracetate. And number D, succinate to fumarate. Which of the following conversions can be termed as a reduction reaction? Again, I will repeat. Your options are number A, malate to oxaloacetate, number B, beta aspartate semialdehyde to homoserine, number C, glyceraldehyde 3-phosphate to 1,3-diphosphoglycerate, and number D, succinate to fumarate. Which of the following conversions can be termed as a reduction reaction? Okay, let me check the chat box now for the last question. Let me check what is the answer given. So number B, no, I am not from Kolkata. I'm from West Bengal, but I'm not from Kolkata. So number B is the given answer for problem number 20. Let me check. Is number B correct answer or not? Yes, very good. Number B is the correct answer. Beta aspartate semialdehyde to homoserine. This conversion can be termed as a reduction reaction. Okay. So there are some examples of oxidation and reduction reactions where the NAD plus and NADP plus they act as a oxidizing or reducing agents. Like the example of reduction reaction is the beta aspartate semialdehyde, which is reduced to homoserine, and here the reducing agent is NADPH. Okay, and the enzyme is the homoserine dehydrogenase. Now this NADP plus and NADPH, these NAD plus and NADP plus, these acts as a oxidizing and reducing agent. Like oxidation reaction, the oxidation of secondary alcohol group to malate to a ketone group is one of the reaction of citric acid cycle, and the oxidizing agent is in this reaction is NAD plus. Again, NAD plus acts as oxidizing agent in enzyme catalyzed reaction for aldehyde group to glyceraldehyde 3-phosphate to an anhydride of carboxylic acid and phosphoric acid. This one, and now I have just given this example like NADPH acts as a reducing agent for the beta aspartate semialdehyde to homoserine, and this reaction is a reduction reaction because as you can see here aldehyde is converted to alcohol and aldehyde to alcohol conversion is the reduction reaction. Okay. So this was the last question of today's session. So today what we did, we discussed about all the things we discussed about the coenzymes and cofactors, many coenzyme cofactors and conversion we discussed and we about. So now okay, so I will share my email ID. Okay. So this is my email ID. If you have any doubt or any problem regarding today's session or overall in any topic up to the course, I mean today is the week 7 live session and we are covering week 8 course content. So we have completed like 8 weeks course content already for in our live session. So if you have any question from the previous weeks also, you can feel free to contact me through this email ID. Okay, and where to find this lecture I have already shown you. So you have to go to the SWAYAM portal here. You will find here weekly contents. Okay, if you go to the weekly contents, you will see that in the last portion there is a problem solving session. If you click on the problem solving session recording, you will be redirected to the page. If you click here, you will redirected to a particular Excel sheet here. This is the portion. This is the YouTube links for the previous classes and today's class which I am recording. I will also upload it in the YouTube and the link I will share here. And this Google Drive folder has all the lectures, all the PPTs up to which we have done till now. Okay, so you can find it out later in tonight for today's lecture and the PPT. So it's up to, till today. I will be closing the session for today. So feel free to contact me through this email ID if you have any question, any queries, or any doubts. And on the next Monday, we will meet for the next our next live session, which will be our week 8 live session, and we will discuss the week 9 course content. Okay, and this type of meeting we will have on every Monday, and you can just save this Google Meet link. Okay, this link is for every Monday class, and we will have 12 total 12 classes. We have completed 7. We have 5 more classes along with a summary class. Okay. So now I will close the session for all. So thank you for joining. We will meet on next Monday, and if you have any doubt, any query, please feel free to contact me through this email ID. Okay. I'm closing the session for today. Thank you all. If you have any doubt, you can let me know right now also. We have 1 or 2 minute time. Otherwise, I will be closing the session. Okay, I'm closing the session. If you have any doubt, please email me. Okay, thank you all for joining. Thank you.